All right, so we're going to be looking at cell responses to osmotic environments. We're going to look at cells with a cell wall, so for instance some bacteria, and cells without cell walls, which would be similar to parasites or us as human beings. We do not have cell walls. So we're going to look at the three different tonicities. We're going to start with isotonic. So this one is going to be isotonic. All right. Iso means the same. And so what isotonic is telling us when we read that about the environment of the fluid inside and outside of the cell, this is telling us that the, let's see, that should be white, that the solute and the water concentration is the same, okay? It is equal inside and outside of the cell. So a solute is going to be those dissolved substances, okay? So let's draw our cell here. This is going to be with a cell wall. So like I said, many bacteria will have a cell wall. And so this is going to be our cell with the cell wall. This is our cell membrane. And then this is our cell wall, okay? So a solute is going to be a dissolved substance. So when you hear the word solute, you want to think something like salt, okay? It does not necessarily always mean salt, but it's those ions, those electrolytes, right? It's a dissolved substance. And I want you to remember when we're looking at salt, if you eat salty popcorn, what do you want? Well, you want to drink water. So salt or solutes, okay, that can be glucose and things like that as well, it's going to draw water to it, okay? So it's going to draw water to it. Let me put our nucleoid region there, okay? And so let's say 6%, and I'm just going to use little dots to represent our percent, okay? So we have 6 inside and 6 outside of the cell. So what that means is water is going to be drawn in and out of the cell. Remember, water travels via osmosis, which means going from a high to low concentration. But it's going to be drawn in and out of the cell at an equal rate, okay? So the rate of diffusion, which osmosis is diffusion of water, is going to again be equal. So there is no net flow. Water still moves in and out, but it's going to move in and out at an equal rate, okay? In and out at an equal rate. So these arrows are indicating the water movement, okay, the rate of diffusion. All right. Okay, so let's go ahead and draw our other cell. Okay, and so here's our other cell, and we're still on isotonic here. All right, here's our nucleus. And again, we're going to have 6% out here and 6% inside the cell. Water concentrations flowing in and out of the cell are going to be equal. And so the cell volume here is going to be stable. The cell volume will remain the same. And this is for both cells. So being stable is good for cells, okay? So this isotonic environment is good for cells. This is ideal. This is what they want. An example here is this is going to be favorable for things like parasites, right? Because we're eukaryotes. Parasites are eukaryotes. We are not having a cell wall. Most parasites are not going to have cell walls, okay? Oop, there should be an S in the word parasite. Sorry about that. Okay, so now we're going to look at changing conditions here, all right? Okay, so now we're going to look at hypotonic. What does the word part hypo mean? Hypo means below. All right, hypo means below. And so here, our solute concentration, sorry about that. So here our solute and our water concentration. Actually, let's start that again. Sorry. Our solute concentration, okay, the solute concentration in the environment, which means outside of the cell, the extracellular environment, is going to be lower than in the cell. Okay, it's going to be lower than in the cell. And our water is going to enter the cell. So let's see what that looks like, okay? All right, so again, we're going to draw our cell. There we go. Oh. With a cell wall, okay? And here I'm going to draw, let's see, I'm going to draw the original one first so you can kind of see it. This was our original cell, okay? This was the isotonic cell. And so this one, if it's going to draw water in, it's going to be larger, right? It's drawing water in, okay? So for our solutes, if it is lower in the environment than it is in the cell, we're going to have 6% in the cell and let's say 3% out of the cell. So there's our 3 solutes. In the intracellular environment inside the cell, here's our little nucleoid region, I forgot to put that in there, is going to be higher. So remember that water follows salt. Of course, and remember again, that's not always sodium. It can be sodium, potassium, calcium. It can be glucose, protein, okay? But it's just a handy way to help you remember it. Water follows salt. And so water is going to be drawn into the cell. And so therefore, the cell will swell, okay? The cell will swell. Now, in this case, the cell wall, sorry. Sorry, I'm trying to keep my colors going here. The cell wall will prevent lysis, which means rupturing. It's going to prevent rupturing. So we're going to have a net flow of water into the cell. Into the cell. All right. So the cell wall here, remember, is important because it prevents the cell from rupturing even though it swells. So this can be a slightly hypotonic environment, can be good for bacteria. So slightly hypo is good for bacteria. All right. So let's look at the hypotonic environment for a cell without a cell wall. All right. So you're going to have the cell here. So let's draw our original. This is going to be our isotonic cell. It's our nucleus. All right. Remember, we have equal rates here going in and out. Oops, that way. All right, so this is going to be our hypotonic for the cell. Well, remember, water is going to be drawn in. All right, so you'll see what's going on here in a minute. So let's draw our solutes. So remember, hypotonic means that the solute concentration in the environment is going to be lower than in the cell. So we have 3% outside of the cell, the extracellular environment. And then we had 6% inside the cell. All right, so we have a higher solute concentration in the cell than outside of the cell. So the environment is hypotonic. Okay, so water then is going to follow the salt and move into the cell. Okay, it's going to move into the cell, and the cell can lyse, it can rupture, and burst. And that is what is going on here. This cell is rupturing or burst. Okay, bursting. And so that would harm the cell, obviously. If the cell ruptures, the cell's probably going to die, right? Okay, so next, we're going to look at a hypertonic environment. If hypo means below, hyper means above. Okay, so let's see, we need green here. So here is our isotonic cell with a cell wall. All right. So this, well, let's go ahead and write this down. So hypertonic above means that the solute concentration in the environment is going to be higher, oops, than in the cell. And so what does that mean? Water is going to leave the cell. All right, so here's our cell wall and our cell membrane. So what did we say? The solute concentration in the environment is higher than in the cell. So in this case, we would have 6% solute out here and perhaps 3 here, okay? And so 6 solutes out there and only 3 in here. So which one is higher? Well, the environment is higher. It is hypertonic, and so our water is going to leave the cell. And as the water leaves the cell, that cell volume is going to change. The membrane is going to pull away. And we call this, because it's the plasma membrane, plasmolysis. Plasmolysis, okay. There will be a net flow out of the cell. So hypotonic, there's a net flow into the cell. Hypertonic, there's a net flow of water out of the cell. So let's look at what happens to a cell that does not have, again, this is hypertonic. So a cell with no, there we go, with no cell wall. All right. So again, here's our isotonic for comparison. All right. Okay, and so this is the cell without the cell wall. Oh, and that's way bigger. Let's change this. Let's say that's the cell, okay, because it shouldn't get bigger. Okay, all right, so remember, hypertonic means that the solute concentration in the environment is higher than in the cell. I forgot our little nucleus here, too. So we'll have 6 outside, 3 inside. What's the net direction of water? is going to go out to the higher concentration, so it's going to go out of the cell. So here, the cell will shrink and distort. The cell can shrink and distort. Spell in there. Okay, this is important because this is a limiting factor for bacteria. They're not able to function. Most cells are not able to function if they shrink and distort. But this is why salt works as a preservative or as an antibacterial type substance. Okay, so think about this. When I was little, we used to, I lived in Louisiana, we had slugs everywhere, we poured salt on them. What happened? Well, the salt killed them. How did it kill them? It pulled the salt out. So this is why they can often use salt to do things like preserve meat, because they put salt all around it, and it is going to be a limiting factor for the bacteria here. Okay, the bacteria are not going to be able to function, or perhaps they will kill them, and therefore your meat is not going to spoil because there's no bacteria there. Okay, so that is hypotonic, hypertonic, and isotonic.